Okay, so this video, today's lecture will be posted on the YouTube channel. Okay, so today we are going to be talking about discussion 9, and these are carbohydrates and nucleic acids. So this stuff is not too bad. There's a lot of rules and a lot of definitions that you have to memorize. I already uploaded to the YouTube channel my previous 1-hour lecture for my previous semester, and we go over the same concepts. Just if you need like different examples, the worksheet is different. So this is our worksheet this time, and then for last semester, it was this one. So if you feel like this is one of those topics where you need more examples to be able to do well on the exam, then go ahead and watch that video as well. Because it gives you, sometimes professors go back to like different worksheets to take problems for like an exam. So the more examples that you see, the more prepared you're going to be. And that's already on the channel. Okay. So I'm going to go slowly today. I did do a lot of notes and annotations. So monosaccharides and disaccharides, you have to know which ones are monosaccharides. So make flashcards. And those are just like single sugars, like 1 glucose is a monosaccharide. 1 fructose is a monosaccharide. 1 mannose, 1 ribulose. And sometimes on an exam, like there will be like multiple choice and they can ask you something about like, you know, select which one is not a monosaccharide. So you have to have them memorized. For disaccharides, we have sucrose, maltose, and lactose. In your discussions, they may also cover other ones. So I would have 2 flashcards, one with all the monosaccharides you encounter and one with all the disaccharides. And that way you won't be thrown off on the exam if you get one of those questions. And then right here for disaccharides, I wrote down what 2 sugars. Disaccharides just means 2 sugars. So we have sucrose, and sucrose is composed of 1 glucose and 1 fructose. So remember, glucose and fructose are monosaccharides. You can combine any 2 to make a disaccharide. Then we have maltose. Maltose is glucose plus glucose. So I like to come up with things that'll help me memorize things better. So I always say, oh, maltose is a twin because it's glucose plus glucose. Lactose is glucose plus galactose. All right. So given the following carbohydrates, number the carbons, draw a box around the carbonyl group. So remember, I like to underline my instructions, carbonyl group, and indicate the carbohydrate if it's an aldose or a ketose. Okay, so we're going to talk about all of that. Now, for these future, like for the rest of the semester, remember at the beginning of the semester, I had you guys memorize your, was it functional groups? Yeah, I told you at least get started and do like a few every week. Hopefully you have, because now they're just going to be throwing them everywhere, all through the final. So hopefully you have most of your functional groups memorized as well. Okay. But where I can, like wherever I can, I have been like telling you guys or reminding you guys what each group is, just in case you haven't memorized it yet, okay? So here we go. We have D-glucose. So whenever you're given one of these molecules, they can ask you on an exam a lot of different questions. So I'm going to explain to you how it works. This is glucose. It has 6 carbons, 1, 2, 3, 4, 5, 6. They can give it to you backwards. So you have to know where carbon number 1 starts. Now, it starts at this aldehyde. So this is the functional group. It's a carbonyl aldehyde. The carbonyl is just this carbon double bonded to oxygen. That is a carbonyl. And if there's also a hydrogen there, then that's a carbonyl aldehyde. So glucose is a carbonyl aldehyde. And that is wherever your carbonyl group is, that is your number one. So it's that easy. 1, 2, 3, 4, 5, 6. So now we know where all of our carbons are, okay? Now, these carbohydrates, they exist in your body in the straight form, but they also exist in the circular form down here. And like most of them, I think like 99% of them actually exist in the circular form because it is more stable. Okay, so that might also be like another one of the answers on a quiz, like which of the following facts is wrong? So you have to know this like fact so you can eliminate. So yeah, about 99% of them are in the circular form. So what happens is that the electron pair on this oxygen will attack. So that's going to be the nucleophile. It's going to attack this carbon on the carbonyl. That carbon is an electrophile. Why? Because it has a dipole. It has a partial positive charge because that oxygen is really negative and it's stealing most of the electrons. So that is why the lone pair on this oxygen will attack this electrophile, carbon number 1, the one that is attached to the carbonyl. And then there's a series of rotations that happen and you end up with this. You have to know how to identify and draw them both ways. So for this chapter, there's going to be a lot of practicing on a whiteboard. So, okay, now there's rules. We're going to learn the rules. You have to know glucose. Memorize glucose first. So notice that there's one hydroxyl on the right, then the next one is on the left, the next one is on the right, and the next one is on the right. So I just memorized that pattern. Glucose is right, left, right, right. Right, left, right, right. That's glucose. So then the next rule is, I'm going to start reading my notes. The hydroxyls on the left will face up. So now we're talking about the circular form. The first rule is that the hydroxyls that you have on the left will face up. And I wrote a little up arrow for you. The ones on the right will face down in the Haworth projection. Sorry, my asthma is really bad today, so I may just have to take little breaks. Okay, so that's the first rule. Or the numbering could be the first rule. The carbon number 1 is at the carbonyl. And then for glucose, we have right, left, right, right for the hydroxyls. And then the ones on the left will face up and the ones on the right will face down. So we're talking down, up, down, down for this circular one. Now D, what are they talking about for D? D-glucose. So the D refers to the chiral carbon that is farthest away from the aldehyde. So this is the aldehyde. And the D is talking about the chiral carbon that is farthest away from that aldehyde. So I'm thinking, okay, this is the farthest carbon from the aldehyde, right? Now, is this a chiral carbon? Who remembers the definition of the word chiral? I figured a lot of you don't, so I went ahead and included it in your notes. Remember, a chiral carbon has 4 different groups attached to it. So this last carbon has 2 hydrogens. You see this H2? It's got a pair of twins. So this means that it is not a chiral carbon because in a chiral carbon, you have no twins. Everyone has to be unique. Okay, so then this guy is not our chiral carbon. So in order to determine whether this is D or L right here, we have to find the chiral carbon that is the farthest away from the aldehyde. So we just start backwards. Now we go to this carbon and we look at it. Does it have any twins? Well, we have an H, then we have this group over here, CH2OH, we have an OH, and then we have this giant group over here. All 4 groups are unique. So this is our chiral carbon that is farthest away from the aldehyde. Okay, so then if the alcohol group here is on the right, this is D. If the alcohol group at that chiral carbon is on the left, then this will be L. And that's it, that's the rule. You got it? So that's why this is D-glucose. Okay, now, if you move this first hydroxyl group to the left, but everything else stays the same, that molecule is going to have a different name, but it is going to be a glucose epimer. Okay, it will be a glucose epimer. It'll just be different in one location. Okay, and so I went ahead and wrote all those like definitions for you all over the place. So let's cover some definitions. Aldose, that's an aldehyde group. At the carbonyl is at the end. What is a ketose? A ketose has a carbonyl on the inside, not at the end. Okay, because sometimes they're gonna just give you a molecule and they won't say anything. They'll just ask you a bunch of questions. So you have to know how to get to the answer based on your definitions. For example, D-glucose, is that an aldehyde or is that a ketone? It's an aldehyde because it's got that aldehyde at the end. It's got the carbonyl at the end. What about fructose? This is fructose right here. Where is the carbonyl group? Yeah, it's inside. So this is a ketose. And that's how you can tell. It's actually pretty easy. So all this stuff is pretty easy as long as you have your definitions memorized and your rules memorized. You can do everything. Okay, let's talk about fructose. So here we have carbon number 1. We still start numbering right here. Carbon number 1, 2. But now our carbonyl is at carbon 2 instead of carbon 1. And then we have carbon number 3, 4, 5, and 6. Now, we have a D. So what does that mean? We go to our chiral carbon that is farthest away from the carbonyl group. And we look at number 6, we have CH2. That is not a chiral carbon because it has twins. So we move up to carbon number 5. This one has a hydrogen, CH2OH, OH, and then this giant group over here. So that is a chiral carbon. It is attached to 4 unique groups. So then we look at its hydroxyl. Is it on the left or is it on the right? If it is on the left, it will be L fructose. If it is on the right, it will be D fructose. Okay, you guys are starting to memorize it? Yeah, all the repetition? Okay, cool. Now, we have D-ribose. So we have 1 carbon, 2 carbons, 3 carbons, 4 carbons, 5 carbons. Okay, so is this a ketose or is it an aldose? It's an aldose because it has an aldehyde group and it's the carbonyl is at the end. That's how we know. So this is an aldose. What about D? Let's go to the chiral carbon that is farthest away from that aldehyde. This one has a twin, so that's not it. And then this guy has H, OH, this group, and then that group. This is our chiral carbon. The alcohol is on the right-hand side, so that's why it is D. Okay, now 2 more definitions. You notice here pyranose and furanose. What does that mean? Okay, so I wrote it for you right here. And these will show up on quizzes and exams and stuff. So a pyranose is a 6-membered ring, but it only has 5 carbons in the ring. The other atom in the ring is an oxygen. A furanose is a 5-membered ring, but it only has 4 carbon atoms in the ring, and 1 is an oxygen. So furanose is a 5-membered ring, and a pyranose is a 6-membered ring. And to each of those, you just take away 1 carbon. So even though a pyranose has 6 members inside of the ring, it only has 5 carbons. One of those will always have to be an oxygen because these are carbohydrates. And so here we see this is D-glucose, right? So here's 1. Okay, let's get into it. Numbering. Numbering here is everything. Everything. You will not pass this test if you do not know how to number. So practice, practice, practice. This guy right here, this is called our anomeric carbon. And he's like the most important little guy. He is the anomeric carbon. That means that he was the carbon that was on the carbonyl group. So this guy over here, that carbon, that's him right here. So he's number 1. Okay. And then you go this way, 2, 3, 4, 5, and then 6. Notice that carbon 6 is not in the circle. Okay. And then let's go back to our definition of a pyranose. Pyranose is a 6-membered ring, but it only has 5 carbons in the ring. Well, this has 1, 2, 3, 4, 5, 6 members inside of the ring, but it only has 5 carbons in the ring, and one of those is oxygen. So this is how a lot of students lose points on an exam, because they'll give you 4 molecules and they'll ask you to choose the pyranose or the furanose. And you'll say, well, there's 6 carbons here though, 1, 2, 3, 4, 5, 6. But they're not, that's not the definition. That's why you have to know your definitions. The definition is that there's 5 carbons inside the ring. They're not asking you how many carbons total, how many carbons are inside the ring. 1, 2, 3, 4, 5. And there's an oxygen. So this is a pyranose. So alpha D glucose is a pyranose. Okay. And now they gave us another variable. Something else just happened. What's new over here? We've got an alpha now. Where'd that come from? How do you figure that out? Well, that's another rule. It's really easy. Where did I write it for you? Okay, so I went ahead and circled both of them, okay? Group number, the alcohol attached to group number 1 to the anomeric carbon, okay? You circle that, and then you go to your 6th carbon and you circle that, okay? If these 2 face in the same direction, this is cis and it is beta. So that's how you know if something is alpha or beta. You just circle the alcohol on group 1, you circle the group 6, and if they're both going in the same direction, that's cis and this is beta. If they face in opposite directions, they are trans and that's alpha. So here you can see that carbon number 6 is facing upwards and the alcohol on the anomeric carbon is facing downwards. So those are opposite. That's trans and this is alpha. Easy? Yeah? Okay, let's look at this guy. Fructose. Okay, so, oh, okay. We haven't talked about that guy yet. Okay, so carbon 1 is known as the anomeric carbon because the alcohol attached to it can face down or up. Most are in the beta form. Okay, so that might be a question that you have, like what form are they normally in? And most of them are in the beta form. So let me read all my notes to you and then we'll talk about this guy. Okay, so in the Haworth projections for D and L, look at carbon 6. If it is up, it is D. If it is down, it is L. Okay. So it's a little different. Remember up here, I taught you D and L, you go to the chiral carbon, that was the rule. The chiral carbon farthest away from the aldehyde, and you look at the alcohol. If it's on the right, it's, and then if it's on the left, it is what it is. But so that's the rule for these guys. But when you're in the circular format, in the Haworth projection, it's different. This rule right here is very important. For D or L, you look at carbon number 6. So that's this carbon, okay? If it is up, it is D. If it is down, it is L. Okay, so that's another rule that you have to memorize for these guys. Okay, now they're also going to ask you on the quiz or the exam about, they'll give you a sugar and then they'll ask you if it's a reducing sugar. Okay, so what does that mean? A reducing sugar can be oxidized, and this includes any sugar that contains a hemiacetyl group. As an undergrad, I had no idea what a hemiacetyl group was because I didn't memorize my functional groups. So an example is lactose. It has a free hemiacetyl group, which can open up to form a free aldehyde group on the second sugar. So I went ahead and copied these notes for you. So this is a hemiacetyl group. So you have an ether plus alcohol on the same carbon. So here we go, ether plus alcohol. Okay, alcohol plus ether. So you just need it on the same carbon, you have your alcohol and then you have an OR. So if your sugar has this stemming from the anomeric carbon, then that is a reducing sugar. I don't want you to lose points for this because this is really easy, right? And we'll go over some examples right now. And then this is an acetyl. This is another very common group that you're going to be seeing a lot. So memorize it. What is an acetyl? Look at this carbon. It has an OR and it has another OR. Those are two ethers on the same carbon. That's an acetyl. So there's acetyl, hemiacetyl, and hemiketyl. Practice drawing those. Okay, so those are examples, but I just have to explain one more thing. Let's see, where are you? Oh, right here, okay. So this is a ketose, right? So for all ketose, they're gonna be a little bit different in the ring. The ring structure is different. So look at carbon number 1. For a ketose, carbon number 1 is not the anomeric carbon anymore because of the way that the molecule folds. So this lone pair of electrons on this hydrogen, it doesn't attack at the end, it attacks here at this electrophile because that's where the carbonyl group is. So then that's why the molecule folds a little bit different to its circular form. So wherever the first carbon is that is attached to the anomeric carbon, that guy's going to be number 1. And he could be up there or he could be down here. But whenever you have a ketose, this is what you're going to see. Okay. So that's carbon number 1. This is carbon number 2. So now carbon number 2, this is our anomeric carbon. So this has not changed. This is still an anomeric carbon. And then 3, 4, 5, and then 6. So how many carbons are inside of this ring? 1, 2, 3, 4. 4 carbons, but this is a 5-membered ring. So that's the definition of a pyranose. Actually, isn't that furanose? Why does it say pyranose? 1, 2, 3, 4, 5, 6. 1, 2, 3. Oh, 1, 2, 3, 4, 5, 6. Yeah, no, that's not a pyranose. So let's go ahead and fix that. Okay, so furanose is a 5-membered ring, but only 4 carbons in the atom. So let's double check. 1, 2, 3, 4, 5-membered ring, 1, 2, 3, 4 carbon atoms. So this is a furanose, okay? And this is a ketose. They're all going to be numbered this way. And then, yeah. All right, so we went over all those definitions, and now we're going to go ahead and do some examples for something that you might see on the exam or on your quizzes. So here's a couple more definitions. Diastereomers can differ at 1 chiral carbon or more. Epimers are diastereomers that differ at only 1 chiral carbon. You have to have those two really memorized because on a test, this can cost you the entire question. Remember, if it's multiple choice, you have 4 choices and you can't be confused about even one of those choices. You have to know your stuff. Okay, so a diastereomer can differ at 1 chiral carbon or 2 chiral carbons or 3 or 4 or 5, but an epimer, everything has to be the same except that there's a difference at only 1 chiral carbon specifically. And you have to, this is why it's so important for you guys to memorize glucose. You have to know how to draw it because then they're gonna ask you, who's an epimer of glucose? So you need to know your definitions. So here's a question. Draw the cyclic form using Haworth perspective formula, that's just the rules, of beta D galactose. So this is what galactose looks like, okay? So it's just, you start off by numbering 1, 2, 3, 4, 5, and then 6. And then you draw its alcohols. Remember, if they were on the right-hand side, they go down. If they were on the left, they go up. From your notes right here, they go down versus up. That's how you know how to draw these. And then it says beta D galactose. So for beta, you go look, if this is alpha or beta, remember you circle the alcohol group that is attached to the anomeric carbon, and then you circle your group 6. They're both going in the same direction. And that's how you know that this is beta. Okay. And then for D, you just go to group 6. If it's going up, then it's D. If it's going down, it would have been L. So it's pretty easy once you know the rules, right? Yeah, okay. So now it's gonna get a little bit more complicated because they're gonna ask you to draw a disaccharide and you have to know how to draw the bonds and everything. So lactose is a disaccharide formed of an O-glycosidic bond. So O-glycosidic bond between carbon 1 of beta D galactose and carbon 4 of D glucose. So all you have to do to start off is just draw galactose and draw glucose. So here is galactose and here is glucose. And then you just start numbering. So this is our number 1, 2, 3, 4, 5, 6. And then 1, 2, 3, 4, 5, 6. Okay, so now it's saying that there's an O-glycosidic bond. Any bond between 2 sugars is going to be a glycosidic bond, okay? And then it's between carbon 1 of this sugar and carbon 4 of the next sugar. So that's how you know where things bond. It's carbon 1 of this sugar and carbon 4 of that sugar, okay? So you just draw your O in the middle. Now, they said the first sugar is beta and it's D. So to determine if this sugar right here is beta or alpha, you have to check out the alcohol on carbon number 1 and realize that this oxygen was an alcohol. So that's the one you're looking for. Now we can see that it is going up. And then we go to carbon number 6 and we see, oh, it's also going up. They're both going in the same direction. That is cis and this is beta. If they had asked you to draw alpha, all you have to do is draw this guy going downwards or draw that guy going downwards. Easy? Okay. Now, they said this guy is D. So in the circular form, to determine if something is L or D, all we have to do is look at group 6. If it's going up, it is D. If it is going down, it is L. Okay. And then we do the same thing for this guy. It says D glucose. So its carbon number 6 goes up. So that's how we know it is D. Easy, right? Okay, lactose intolerance is caused by the absence of the enzyme lactase. So the enzyme is lactase. We don't have lactase, and that's why we have lactose intolerance. Explain which bond would lactase break. Name the resulting product after digestion of lactose with lactase. And why The undigested lactose causes the typical digestive symptoms of lactose intolerance. So lactase breaks the O-glycosidic bond between carbon 1 of beta D-galactose and carbon 4 of D-glucose. So if you look up here, we have glucose and we have galactose. We actually have exactly what they're talking about. It's beta D-galactose and D-glucose. So this is the bond that they're talking about. So if you like drinking milk, like I love milk, I've always liked it. So I have no issues with lactose intolerance. And that means that whenever I drink milk, these are the two sugars. This is the disaccharide that is inside of milk. And I have the enzyme, like my body makes the enzyme that goes in and breaks down this bond. And that is why I don't have issues. But for like my cousin who's lactose intolerant, her body does not make this enzyme. So she cannot break this bond ever. So what happens? So the resulting products are galactose and glucose. That's just like this sugar plus this sugar. If you have the enzyme and the bond breaks, you're just left with the two individual sugars. Lactose and other disaccharides can't be absorbed in the small intestine. So undigested lactose goes to the large intestine where it increases osmolarity. And as a consequence, water movement is favored towards the intestine, causing loose stools. Also, lactose is consumed by gut bacteria. So if you don't have the enzyme to digest it, the bacteria, they do have the enzyme. So they're going to go ahead and digest it. And that's not good because that's going to release a lot of gas. So that's why people who are lactose intolerant, if they do have this disaccharide, they end up with those symptoms, which are really uncomfortable for them. Okay, so that's how that works. Structure of DNA and RNA. Okay, so I made a few notes for you here. Again, you just have to know how to draw everything. So practice on a whiteboard and don't wait till the last minute because memorizing all this stuff takes a long time. I think you know that by now. So you have to draw the nucleotide, the 4 nucleotide monophosphates that form DNA and indicate nitrogenous base, pentose, indicate name of the pentose in the DNA, phosphate group, nucleotide, name the single letter, symbol, classification, purine versus pyrimidine, and number all the carbons. Okay, so you have to know everything. That was a lot of definitions, a lot of vocabulary words. If you don't know some of them, you need to know all of them, okay? So I went ahead and wrote some notes for you here. Nucleotides are composed of 3 elements. They have a sugar. The sugar can either be ribose or deoxyribose. Remember, deoxyribose is just missing the 2' hydroxyl, and we'll go over the numbers in a little bit. The phosphate group, and then you have a nitrogenous base. So that is a nucleotide. That is the definition of a nucleotide. You have a sugar, you have a phosphate, and you have a nitrogenous base. So this is the phosphate, this is the sugar, and this is the nitrogenous base. So the nitrogenous base can be ATGC for DNA, or it could be AUGC for RNA, okay? So then these are the ones that we have. Wait, let's go over the numbering. So I put a picture in here for you. Yes, okay, you have to know how they're numbered. So remember your phosphate group is right here. You have your nitrogenous base. It could be ATGC or AUGC, depending on if you have DNA or RNA. And then we have your 5-carbon sugar. It's called a pentose. 5-carbon sugar is a pentose because they could trick you on an exam with a multiple choice question and they can say, oh, the sugar in there is a hexose or it's a 6-carbon sugar. And you have to know, no, that's not true. It's a 5-carbon sugar and it is a pentose. And then this is how you number them. So this is carbon number 1, and it's called prime, 2 prime, 3 prime, 4 prime, and 5 prime. The reason that the carbons on the sugar are labeled with prime is because the base was labeled with the original numbers, just 1, 2, 3, 4, 5. So the sugar has to be 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. That's why. But it's important to know the numbers because that's how it'll help you understand other definitions. For example, this, does anybody know if this is RNA or DNA just by looking at this? Because you should. Anybody have any guesses? Okay, so I'm going to tell you it's RNA. How do I know that this is RNA? This guy right here. This is RNA because there is a 2 prime hydroxyl. So sometimes they'll just give you a picture and ask you a bunch of questions, and this could be one of the questions that they ask you. Is this DNA or RNA? This is RNA because its pentose sugar has a 2 prime hydroxyl. But for DNA, there is no hydroxyl here. That's why it's called deoxyribonucleic acid. Do you guys know that? Deoxy. D means missing. Oxy means oxygen. So deoxy, missing that oxygen there. Okay. So pyrimidines, they only have 1 circular structure. And you start labeling here at the nitrogen. That's number 1, 2, 3, 4, 5, 6. These are pyrimidines. And then for the purines, they have the 2-membered ring. Okay, so practice drawing these, practice numbering them. And you have to know who is who. For example, pyrimidines, I wrote it for you here, C-U-T, cut. That's how I remember it. It sounds scary, but it's like, that's what it spells out. So I'll remember that forever, cut. Cut are pyrimidines. So what is cut? It's C, U, and where did T go? I don't know where T is at. So it would be T also. So that's cut. Thymine, cytidine, and uridine. Uracil. So then AG, so if you look here at our notes, AG right there, AG is purines. So you guys see that? I try to label this as much as possible. All you have to do is memorize it and you should be able to answer any questions, okay? So let's keep going. We have another 12 minutes. So sometimes on an exam, especially now that it's in person, they might ask you to draw the backbone. And then they might give you specific nitrogenous bases. So they might tell you draw TTGA. And then they might ask you questions like, is that RNA or is that DNA? Well, there's a T, so it has to be DNA. If you have a U, then you know that, oh, that's RNA for sure. Okay, so it always starts with 5 prime and it ends at 3 prime. And that's your phosphate group. This little circle here is your phosphate group. This is your pentose 5-carbon sugar. And then this is your nitrogenous base. And you'll notice that GC has 3 hydrogen bonds. So the bonds between GC is stronger and it's harder to cut. It's harder to cleave that bond because it is stronger. So if you're using heat as an energy source to break the bonds, you would need more heat to break the GC bond than you would to break an AT bond because that's held together by only 2 hydrogen bonds. So if I give you 2 nucleotides in the lab and I tell you, oh, how much sugar or how much heat do you need to break down the bonds in this one versus this one? If this one is full of GCs, there's 1,000 GC bonds, you're going to need a lot more higher temperature. But if I give you one that has a lot of ATs, then you don't need that much temperature, right? So you have to know that. You have to be able to like go full circle with these concepts. So a new nucleotide will be added here at the 3' hydroxyl group. So I've seen that question being asked on quizzes and exams for years. Professors love that. Where will a new nucleotide be added? At the 3' hydroxyl. That's where. The hydroxyl group of the carbon 3 and the phosphate group of the new nucleotide will react to form a new phosphodiester bond. So you have to know about phosphodiester bonds. And then this is the sequence, it's GTCA. Watson and Crick deducted the structure of DNA, a double helix formed by 2 antiparallel strands where the nitrogenous bases are paired in the interior of the helix. On the above figure, draw the complementary DNA strand for the above sequence, GTCA, and draw the hydrogen bonds formed between complementary nitrogenous bases. Okay, so we did that. Imagine that the above DNA sequence contains the genetic information for the production of functional protein. Messenger RNA acts as an intermediate to carry the genetic information from the nucleus of the cell, where the DNA is, to the ribosomes in the cytoplasm, where the protein is made. How does the structure of messenger RNA differ from the structure of DNA? Write the messenger RNA sequence that will result from the transcription of GTCA sequence above. So messenger RNA consists of a single strand of RNA. So that's one strand. That's one way in which they're different. So they might give you a multiple choice question and ask you which of the following applies to RNA or which of the following applies to DNA. And then they might just give you a bunch of sentences, and you have to be able to like say, oh, A, B, and C are DNA, or just A and B are DNA. Like you really have to know your stuff. So RNA is a single strand, and it tends to assume a right-handed helical conformation dominated by base stacking interactions and regions of self-complementarity. Sequences can generate more complex structures. So RNA can bind with itself. Self-complementary, that's what it's saying. The messenger RNA sequence from 5' GTCA to 3' would be CAGU 5'. RNA has U instead of T. Yeah, so if they ask you to get the sequence complementary, you just have to go, well, G binds with C, T binds with U, no, T binds with A. Yeah, C binds with G, and A binds with T, but they want the RNA, so you have to put U. Okay, so pay attention. So DNA can undergo reversible denaturation, for example, by heat. So this is what I was talking about. There's temperature right here at the bottom and denaturation on top. So I've seen like this actual question on an exam before, so pay attention. On the right-hand side, you have temperature, on the x-axis. So we have from 75 to 85. So low temperature, high temperature, and denaturation. So they can ask you like for B, this blue line is gonna be the one that has a lot of ATs, because notice that the melting temperature was only at like 82 degrees. But this red one required a higher temperature to get the melting temperature. So this red one is gonna have more GCs. That makes sense, right? Easy. You're just able to do it with a graph. And then the last thing I wanted to show you is, I copied this over for you guys. So it won't be in your original key. A lot of things won't be on your key. So these are like the most common things that might come up on your exams. So here is D-glucose. Remember the alcohols, right, left, right, right. And then mannose, I underline this 1-hydroxyl because mannose is a carbon 2 epimer of glucose. So you guys see, wait. I don't know why it's not letting me unclick. Okay, it's not letting me. Okay, so mannose is a carbon 2 epimer of glucose, and it might show up on an exam. So that means that carbon number 2 is the only one that is different, because in glucose, that alcohol is on the right, but in mannose, that alcohol is on the left. And then galactose is another epimer of glucose. So glucose is super important. It's the star of the show. And you have to know its epimers as well. So this is the carbon 4 epimer of glucose because 1, 2, 3, 4, it is different at carbon number 4 and only carbon number 4. Do we know what that is? Huh? Yeah, it sounds like it, right? It doesn't sound like a fire alarm. Yeah. I've never heard it. Oh my gosh. That's so crazy. Okay, and that basically concludes today's discussion. It's just a lot of memorizing and a lot of